today. Let me just maybe say a few words. We have 2 lectures in the morning, and then we have kind of a virtual lab. We are still, where are we? We are still dealing with kind of developmental biology. You have not heard that much yet about how organs develop and things. So we have rather looked at methods, how you use genetics to ask biological questions. You want to remove DBP somewhere in the animal, you have to have a DBP mutant. Maybe it's an enhancer mutant, lacks an enhancer for the midgut. You want to ask whether thick vein is important. You take a thick vein mutant. Actually, the genes were cloned starting with these mutants. So genetics and development are intricately linked. And the first lecture is a little bit about genetics, about CRISPR. You know most of the things, but I want to put this into a context so that for the second lecture, which is about a completely different approach to do developmental biology, you sure have never heard about, most of you. So the first lecture is kind of to prepare you. What can we do with genetics? What's the problem? Where do we need new methods? Then Shinya, Ambizione Fellow in the lab, will talk to you about wing development. So we will go a little bit more into this wing development field with the new methods that I will present this morning. And then in the afternoon, Gustavo will be back. Martin will be back to discuss some genetics. And you also can continue to prepare your paper. You will not hear that much about developmental biology afterwards anymore, so I think it's a good time for these 2 lectures. So let me share the screen. I don't know, it's kind of funny to sit here all alone and talk and have no clue whether there is somebody at the other end, because I don't hear anything, but I guess somebody's there. So this first part is the shorter part. It's in German. I'm sorry for this, but it doesn't really matter. I just want to make a few points that you hopefully remember in the future. Details one never remembers. Bigger points, concepts might be easier. So CRISPR, you have heard about CRISPR. It's a method to genetically modify genomes. And I'd like to go a little bit into this and where we started and where we are now and what comes next, maybe, to prepare the territory for these completely new methods in the second lecture. So this is a general lecture. I've taken it from, I have given it for another purpose, but I think it suits very well. If I can, why can I not do that? Let me go back. I should be able to share screen. Can you see now is the whole screen with the PowerPoint? Alessandra? Okay, you seem to be able to see it. All good? So let me know if you tell me after half an hour that you couldn't see anything. It would be a pity. So let's start. What do we, in developmental biology, what is the aim? What we would like to understand is how animals like this, flies, or it might be a plant, it might be a mouse, it might also be a human being, how they develop. For me, in the past several hundred years, of course, it was very important and key to identify DNA as the material that carries heredity. That's absolutely no doubt. Watson and Crick, the structure of DNA. But I was most impressed by Schleiden and Schwann when they discovered that the basic unit of each and every organism, whether this be plant, microorganism, vertebrate, human, is the cell. This is the basic unit. And cells are different in prokaryotes and in eukaryotes. This is a typical eukaryotic cell. This is the basic unit. It's the It's the functioning of these cells and the cooperation between these cells, the proper placement of distinct cell types that makes animals what they are. So it's the basic unit is really the cell. What we need to understand is the cell, and then how cells communicate, interact. Of course, there is extracellular matrix. So it is very important to understand how a cell functions. How does a cell divide? A lot of initial cell biology was put on cell division. Cancer is sort of outrageous, non-correct or non-proper cell division. So there was put a lot of effort into understanding cell division, and we know now a lot about cell divisions, but we don't really know how a cell knows how many times it has to divide to make an organism of the appropriate size, for example. We know virtually nothing about this. So cell division, yes, we know the basics a bit of the job for cells to divide, but how and why our eyes get exactly the size they do, why the left and right eye are the same size, why the left and right wing of the fly are the same size, why is a fly not twice as big or half the size, we know very little or virtually nothing about it. So yes, we know how a cell divides, but the organismic control of all of this is not so well known. How do cells communicate? That's what is the major focus of this lecture. We have seen DVP, it can disperse. We'll learn more about it. How far can such a molecule go? Can it go from the head to the tail, or is it only short range? How do cells communicate over distances? And how do they change their properties? This is mostly by regulating the genes in different ways. There is one cell to start with. The genome has one property, this first nucleus. Then you generate several cells, each has a nucleus, and then you start to make the nuclei transcribe different genes. And as you go, the cells become more and more different and more divergent, and they end up in brain cells and gut cells. So what we in the lab have studied, we have studied a lot of tracheal development. You might, you will not see that much about it. It's a bit similar to the lung, but actually it's much more similar, as it turns out now, to blood vessels, and you will hear more about blood vessels. This we study in zebrafish. Tracheal development we study in flies. It turns out there are a lot of similarities, but there are also differences. This has been the main topic of my lab. And of course, people use different model systems, and this is important. Each model system has an advantage. C. elegans is very small. It's not very cute at first sight because it's kind of a worm. The first, second, third larval stage, they all look a little bit the same, but it's simple. 958 cells at the end of larval development, and so on. So every cell has, you know exactly where every cell comes from. So genetics very fast, generation time very fast. Fly is a bit bigger. It has things you can recognize. It has legs. We have legs. These larvae don't have legs. So leg development in C. elegans is not so easy to study. You can do this in flies. So as you go a bit higher, you get more and more things that we know of. The vertebrates, they have a vertebrate. Zebrafish, it has a vascular system. Flies don't have a vascular system. And then we come to mouse, and ultimately we come to human, and we'll see that a bit with organoids. So many different models. Most of them were used, or many of them were used for genetic approaches. First, Xenopus was used for transplantation. Yesterday in a course, somebody said we could transplant cells in Drosophila and see cells that make DBP to another place, and then see what that does. Drosophila is small, so and is the larvae, the immunological disc is inside the larvae, so it's difficult to do cell transplantation. Has been done, of course. People have transplanted embryonic cells from one segment to the other in 75 and realized that they're specified in the embryo to which segment they belong. If you transplant them, you need to transplant several cells. They do the same thing. So different model systems. Nowadays, genetics is, I would say, the most prominent approach to study development, organogenesis, all of these things. Not anymore transplantation. Of course, genetics, if this is very important, then we need to know why and how. And this is, for me, really the best example of why genetics are so prominent and are so interesting to use, because if you have the same genomes, then you look the same. You can have more muscles, less muscle, you can have a different haircut, but the physiognomy of the nose, of the lips, of the ears, and of the eyebrows and everything, it's in the genes. It's all in the genes. And if we would understand the genome, really, we could put it into a computer and then ask, what does this person look like under normal circumstances? 10 years old. What's the skull? What's the nose? What are the bone shapes? We should be able to read that from the genome, because same genome, same type. Of course, we are very far away from this, but the same holds for internal organs. If you know the genome, you should be able to read it. And of course, we don't. We know a few genes. Very few genes. There are big enhancer regions we don't understand. And when a gene is expressed and how long it's expressed and how many cells divide in what direction, this is eventually what gives rise to these phenotypes. But it's in the genes. It is in the genes. The cells do the job. The genes control what this kind of job the cells do. So the information for the body plan is in here, in this DNA. This is clear. The DNA is in the cell, in the nucleus, and controls whatever is here. All of the things you see here, all of the cell communication stuff is eventually at some point controlled here, but the effectors are all out here. DNA doesn't do anything. DNA is the most conservative molecule. All it wants to do is to be replicated in exactly the same way so that you don't have errors and the next cell gets the same genetic complement. So it's the most interesting molecule, but it's a molecule that doesn't do anything. It's a bit exaggerated, of course. The proteins go on the molecule and transcribe RNA from it, replicate it. The proteins do something with the DNA. The DNA is conservative. Just do exactly the same. Get replicated by the protein machinery and do the same. But the letters in here are important. And if we want to study development, then we need to change the code in here. Or then at least one very good approach is to start study things in here. Change this code, alter it, these different base pairs. Of course, in genetic analysis, loads of mutations. I don't know how many. There are 25,000 genes maybe in flies. There must be many more mutations isolated than 25,000, because in every gene you can have null mutations in the coding region, stop codons. You can have gain-of-function mutants, different amino acid that change the protein. You can have a deletion of an enhancer, which makes that the gene is expressed in all tissues but one. There are many, many different type of mutations for each gene, and for many of these genes, many mutations have been identified. For DBP, there must be hundreds of mutations that have been identified. Some in the protein, many in DNA sequences that control the expression of the gene. So this is endless. You can identify, you need to identify thousands of mutations. And of course, you want to do that in a design manner. And EMS, what Janni Nüsslein did, feeding chemicals to flies and look for altered development, concluding that there must be an altered gene, and then you'll go all the way back. The genomes are sequenced now. So we can go right into the genome and change the sequence, duplicate the sequence. With this genomic sequencing, which was, of course, not present when I started. The first genes were starting to be cloned in flies 30 years ago. So in these 30 years was an absolute revolution. That's an important point. 30 years might seem long. It's not so long. So only 2000, it's 20 years ago, the fly genome was sequenced. And the fly genome was only sequenced because Craig Venter proposed that to sequence the human genome, you don't have to do it in any organized way. You just sequence bits and pieces as much as you can, and then let the computer align these sequences. And then you confirm with other sequences that these alignments are correct. And he said, this is how myself, with my lab, I can sequence the human genome by myself. And people were laughing at him, and because the Europeans wanted to do it the European way, England, chromosome X, and Switzerland said, we don't sequence, this is boring, this is technical stuff. We don't know sequencing project was ever supported by the National Science Foundation. This was sort of stupid work, not stupid work, but it was just work. It wasn't hypothesis-based. Gerald Rubin Hurd, the fly person who developed the P elements in flies, he of course knew how important genetics is, how important the sequence is. And he went to see Craig Venter and said, look, if you're right, why don't you help us to sequence the fly genome, put it together, because we know quite a bit about how it's put together. And if you succeed, then you know that your approach works. If your data and the data we already have by classical methods overlay. So within a few months, Cellular Genomics, they sequenced, Craig Venter's firm sequenced the fly genome, put it together, and then it was published in 2000, before the human genome was published. So since 2000, we have a relatively good genome. Now we have a very good genome. Of course, repetitive sequences are a problem. So the human genome is not that good in that sense. There are, of course, a lot of repetitive sequences you don't really know. But Craig Venter sequenced his own genome. And then, of course, you have 2 copies of each strand. So that's, you want to know which copy is which. Otherwise, you assemble a genome that doesn't even exist. You have chromosomes that are mixes of your chromosomes that don't exist that way in your body. And then you have a problem too. So it's not so easy to sequence genomes, put them together the right way. But we are getting there. And if you are there, then you also know if I change this base pair, this is probably the consequence. This is probably what I do. Many, many genomes are sequenced. You can sequence anything. The sequencing capacity is outrageously high nowadays. You could sequence your own genome for a few $100. You'll probably get it in a few days. I wanted to do it. I've never really done it. Switzerland is not very DNA sequencing friendly. I would find it kind of interesting, but I can also live without having a look at my genome. I would find it scientifically interesting. There are problems, ethical problems, and so on. That's clear. So genomes, once you know them, or even when we didn't know them, we won't always want them to change them, because it's written in there what we are, the way we develop. A lot of things are written in there. So you want to have it in research, applied science, medicine. And now, of course, we have methods that are fantastic. You as student, you have never even known the difficult times of DNA manipulations, because you came in when this method was already existing. So you came in when the end dream kind of how to modify genomes was reached. But this was a long way, and I briefly want to go through this way, how that goes. So what do you need to manipulate the genome? If you don't take chemicals, X-ray, substances like this, which just affect base pairs, and then base pairs are excised and misreplaced, and then you have a mutation. If you really want to say, I know this is my protein, this is an arginine, I want to put another amino acid there, in the genome, only one nucleotide change, one amino acid change, and ask what this is. You need to be able to cut DNA, because cutting tells the cell, I start to repair where this cut is. And then you can control how you repair this. This is the whole idea of how you manipulate genomes. It's recognize a sequence, cut at this sequence, and then put another sequence in that place. And this is for sentimental reasons. One of the first persons who actually learned or had established mechanisms that bacteria use to cut DNA was Werner Arber. He is at the Biocenter. He wasn't at the Biocenter when we discovered these things. He did his work in Geneva. And what he found, no details, is that there in bacteria, there are enzymes that cut certain sequences, 6 base pairs, G, A, A, T, T, C. He had other enzymes, not these enzymes. His enzymes cut at a distance from the recognition side of the enzyme, but this is a classical restriction enzyme we use. So this was the first time people realized there are proteins that cut DNA at specific places. Now, of course, if you have a 6 cutter here, you cannot modify the human genome with a 6 cutter, because it will cut once every 1,000 base pairs, so it will induce about 1 million cuts or 10 million cuts in the genome, and then you have 10 million maybe good and bad repairs. I mean, there's no way to do it. But it was a beginning. That was 1975 or something like that. So 55 years ago. 55 years ago, first methods discovered, first mechanisms discovered how to manipulate genomes. Then when I was in the Gehring lab, the first DNA binding domain from a vertebrate protein, a Drosophila or vertebrate, invertebrate, vertebrate, but not bacteria, was cloned. And the structure of the first transcription factor was Antennapedia. That protein was purified here by Martin Müller, the guy who see again this afternoon. He purified this protein in his PhD. Very difficult job. No strains that would highly amplify proteins. So he had to purify the hard biochemical way as a student. He succeeded. Gehring asked the people at the X-ray people in the biocenter to solve the structure. They didn't succeed. So he went to Kurt Wüthrich, who had developed NMR as a completely new method to determine proteins, but they had never determined any protein structure. So Gehring said, maybe you can do this, and Antennapedia homodomain. They did it, and then that was published maybe 1990, so we're a bit further back. We knew the first time the structure of a DNA binding domain, Antennapedia homodomain, and then quickly afterwards, people came up with zinc fingers. Zinc fingers were other DNA binding domains. They bind like with a finger in the DNA. And I remember very well, there was a guy at the Biocenter who already told me in 93 that his dream is to take synthetic zinc fingers, knowing that what each finger cuts, a 3-nucleotide, TTG, ATG, or whatever. So you have different fingers that cut different sequences. You synthetically put them together, and they would recognize these zinc fingers, artificial zinc fingers, would recognize a sequence. You add a nuclease to it, and then you can generate a synthetic cutter that is flanked by 2 sequences for which you have made artificial zinc fingers. That was maybe 93 when he told me this. They succeeded. They got it to work in 2008. The first zinc finger-based nucleases were sold, I think, on the market. We also bought some of these zinc fingers to modify a gene in zebrafish. Cost 25,000 to get this. One nuclease like this. It actually worked. We could modify the locus we wanted to modify by having sequences sent to the company, and they screen the genome for sequences in our locus and send us the best zinc fingers. It worked. So this was clear. Wow, we can modify genomes in a sequence-specific fashion by these synthetic zinc fingers. They would have won the Nobel Prize if things would have stayed at that price. I mean, this is a big change from a restriction enzyme that cuts a 6 base pair everywhere in the genome. Here, you could go quite specifically. There were, of course, off-target effects, but you follow your mutation and hope that the mutations on the other chromosomes get lost as you do crosses. So you have to cross out your mutation or cross out the second side mutation. Fantastic. 2011, suddenly, somebody published something like this, Talon. An easier way to recognize not 3 nucleotides by 1 zinc finger, but 1 nucleotide by 1 module. So there was a module would recognize A, a module that would recognize G, T, C. So you go to your sequence, and then you just align these binders. If your sequence is ATGC, you align the A binder, G binder, T, C binder as a protein. They will fold properly, and they will make such a wheel here, and they will go and recognize your sequence that you want to recognize. You add a nuclease, and then you have a very nice enzyme. 25,000 per zinc finger you paid for a company. This was for free. This is for free. You do it yourself in the lab. You take your sequence, and then you align by PCR. So you synthesize these small fingers, 60 amino acids of each. So 10 base pairs make 600 nucleotides. That is easy. You send it to a company. They send you the DNA back. You make the protein, or you inject it into zebrafish. The protein is made. it cuts, and then you analyze what happens at these cuts. Sometimes they repair properly, no mutation, improperly. So 2011, zinc fingers are not used anymore. People use talent. Fantastic, great, every student can do it. And of course, we thought this is it now. We are there. That's the best method to modify the genome. And then came the surprise, this CRISPR-Cas method. Of course, when you go back to this, if you look at that, proteins are used to bring this nuclease to a certain place. And the specificity of a protein for DNA is, of course, never exquisite. Biology is all about specificity, as I always said. But proteins, they go on and off. They go somewhere. If they just go a little bit and it cuts, the protein might be more stable here at this real sequence, but it might also bind here less stably, but you still might get a cut. So the complete specificity is not to be reached. If you would have an RNA molecule, I mean, RNA and DNA has to prepare perfectly well. Otherwise, you get mutations. You don't want that. Nature has done everything to make DNA either pair properly, or if not properly paired, to be repaired. So the exquisite specificity comes from DNA strand interaction and from DNA RNA interaction. And as it turned out, CRISPR was a method that one, in retrospect, if you would have been a student in 2010, you could have said, wow, I'm clever. The things were there. You could say RNA is much better to recognize DNA. I had a hairpin. I let a protein bind, the enzyme that cuts DNA bind to this hairpin, and I generate a synthetic nuclease. Nobody had the idea, I think. Maybe somebody had. But nature did it. And that's what they identified in bacteria. A lot of work, because they realized that phages have sequences that are then incorporated into the genome of E. coli. Oh, wow, how does that work? And so on. And what they came up with is this here. Bacteria have enzymes, cutting enzymes, DNA enzymes that cut DNA, and they're brought to this specific place of DNA by binding to a hairpin RNA molecule. And this hairpin RNA molecule has sort of an extension which carries sequences. If it carries sequences complementary to the genome, the RNA will pair here, the hairpin will be here, the protein binds to this hairpin and then cuts. You could have done this synthetically. You could have said, that's probably what I want to do. And of course, people now, they want to make synthetic enzymes. This is a huge the protein binding to this RNA hairpin here is about 1,000 amino acids, I think. So it's very big. It's very difficult to bring into cells for human therapy or whatever, 1,000 amino acid protein. So people try to make it shorter so they can put it easily on viruses and infect cells. It's a lot of work going on in the CRISPR field to make this even much better, using exactly the same principle. Now, I'm not sure that any of you or me or can think of an even better system. Full specificity, best specificity here. Yes, smaller enzyme would be good because you want to be. So, but conceptually, is there more? Of course, we didn't think that there is anything more after this, after zinc fingers. We knew, people knew that after restriction enzymes, you immediately recognize that let's make these enzymes more specific. So the idea from restriction enzyme to this was logic. People wanted to do it. People needed DNA binding proteins. They were not discovered in 78 for vertebrates and so on. So there is a long way to go, but it was clear what to do. This was discovery. Nature had actually thought about it too, how to cut DNA specifically, and had developed its own system in bacteria, not in vertebrates. And then this was again complete discovery. We didn't know. But nobody actually thought, let's make zinc fingers much better. Let's take RNA molecules. In retrospect, this is very surprising. Why didn't people think about it? It's so logic once you've seen it. But nobody had the idea. Synthetic biology field is relatively new. more than 10 years, of course, probably 20 years old, this synthetic biology field. People did a lot of things, synthetic proteins, but nobody thought about something like this. But now we have it. We can do human cells. You can inject the protein. You could inject it into eggs. Of course, human cells, it's always the ethic things. You can study human cells, fibroblasts, and so on, ES cells, stem cells, iPS cells, and so on. So you can do a lot of things on human cells because you have CRISPR. Before, you couldn't do genetics with cells. There's no way. We could target the genome very easily with these zinc finger nucleases. It was very expensive, but it was feasible. It worked for us, but it was very expensive. So if it costs 25,000, not many labs can afford to do 10 genes, 250,000. Now it's cheap. You only need to order a very small piece of DNA, and then you do some cloning and inject, and you can do a lot of things. And you can, of course, work on bacteria, plants. The methods are there because we know now where to cut, and then we can add a template after the cut and repair according to this template. We can just cut and hope that nature makes deletions or insertions, or we can tell the organism by injecting a template of what mutation we want. And then, of course, you see afterwards whether this is correct. And you can add 2 of them, you can add 3 of them. So you make deletions, inversions. It never ends. There are thousands of manipulations that people want to do, need to do, with the same aim as I started off. How does the genome work? How can we better understand the genome? We know so little about it. We know some areas code for proteins, and we think maybe these stretches of DNA, that's the only thing they do. They're there to later give rise to the RNA, then gives rise to the protein and the phenotype. But these intergenic regions, the enhancers, the control, the temporal control, which makes some animals bigger, some animals smaller, small changes that have big effects, maybe only on size, not on overall shape. All of these things need to be done. So now we have simple methods. As Jennifer Doudna said, you're only limited by your imagination. What people want to do. So you can go now to any organism you want, and you're allowed to, hopefully, or I mean, sometimes it's useful to do it, sometimes it's interesting, and sometimes it's stupid to do it. Different people have different ideas. Dogs have always been used for a lot of genetic crossing, and all kinds of dogs, weird looking dogs, long hair, long ears, and who knows what have been made. Should people use CRISPR? Certainly not in my opinion. I'm not really sure they do, or commercial use of CRISPR in dogs, would that be allowed? I don't know, not in Switzerland. You can clone dogs, your own dog, before he dies. You can take cells and then clone it and get the same dog. You can do that in South Korea. So there are a lot of things one can do. The methods are there, and for us, they're mostly very useful to do basic science. And I think basic science helps to understand a lot. Now it's very nice to see that science is actually, if we can fight the virus, then it's only by science. Of course, we can fight it by washing hands, so that's very important. But otherwise, to understand what is here, this invisible enemy, as Trump would say it, we know what this invisible enemy is. It has been sequenced. By sequencing it, it is clear who infected whom and how these transmission chains work. So I love basic science for a lot of things. And usually people try to do good things with science. So to understand the genome and why the genome is so prominent in all of these nitty-gritty details of our body plan and of the body plan of each individual, methods like CRISPR are important, because if we want to get detailed information, we need excellent methods that cut once in the entire genome only. Nowadays, the sequencing capacity is so big that if you change a cell, you just sequence its entire genome after and ask whether there is really only one change. Again, things that were completely unthinkable a few years ago. Just do quickly the whole genome in order to see whether you have only one base pair change. I mean, I spent a few years to sequence a few hundred base pairs in the good old days. Now you do the control experiment is let's sequence the genome. Very different types. That's what I wanted to say. Now you can do genetic cell biology. Very interesting. You can go to any animal model you want, butterflies, all kinds of interesting things. You can analyze multiple genes, and this is phenotypes are made. Our phenotypes too are made by how genes work together. It's not that this gene is for A, this is for B, this is for C. So it's multigenic. All of these things are multigenic. A bit more DBP and a bit longer DBP will alter the body plan. People with a big nose, do they have a little bit longer DBP? Maybe they have a little bit longer DBP and a bit more receptors and a bit more bit less down regulation via that. So it's multigenic. And we need to isolate the effect of one mutation or two mutations or three mutations in the context of the whole organism. Do you need these three to do this? Is one prominent and so on? So multigenic, only possible now, wasn't possible before. So there is enormous possibilities for discoveries. That's good, but also enormous possibilities for synthetic biology to introduce new things into genomes at the right place, so that they're properly regulated. So the CRISPR revolution, and this is keep it in mind. I think it's one of the biggest revolutions that has ever happened in the field of biology. We take it for granted to use it, but it's only a few years old. It's cheap, important for the second lecture. It's fast. Fast is always important. Zinc fingers were not cheap. They were as fast. They were not as efficient. This method is efficient, and people work extensively on the efficiency, make enzymes that are more efficient, smaller, have better properties. We have synthetic biology field. If you give them something, they always try to improve it because they know how to handle molecules now. It works in any size organism. That's also, it's not a method that is, I mean, if you P elements, you need transposable elements, characterized transposable elements in flies. If you want to go to another organism, you need to first find a P and transposable element in this species to use the same approach. Here, you inject. You don't need a specific approach. So this method has all the things it takes: cheap, fast, efficient, any size organism, no all you need is the genome of this organism, and then you can go. The Nobel Prize, as Martin said, they still didn't get. There is a bit of fight on patents, of course, so, but they got all the other prizes, of course, and they will get the Nobel Prize, I hope. It's only limited by your imagination. New applications are published on a daily basis. If you go in the break, which comes now to PubMed and make CRISPR, you will get thousands of papers, and you will find very interesting things that have been published yesterday or the day before or a week before. So so you can do organ biology, you can do work with patient-derived organs and manipulate them. Maybe we'll find out what the coronavirus receptor is. I think we already know a little bit about it, but you can test that then in an organ model very easily. Mutate the receptor, are cells still infected, and so on. You can do lots of things, and now people, of course, and this is not sort of the end of this, but will lead to the next. Of course, I didn't think about it, but a lot of people, when they saw, oh wow, we have a RNA-based method to modify DNA, then you say, why can't we have an RNA-based method to modify RNA? To modify an RNA molecule in the cytoplasm. So then we get much closer to RNA. I mean, we have the flow of information is DNA, RNA, protein. So DNA, we're done, I say now. Who knows? Maybe in 10 years we have a method that we've never thought about and is even more clever. But the wish list for how we want to manipulate DNA is done. We want to modify, sequence specifically, fast, cheap, blah, blah, blah. We have it. So I would say, I don't see any new wishful thing in there. Of course, triple mutations and inversions and translocations, all the same way, okay, but we can actually do it. I don't see how we can move even further in this DNA editing field. RNA editing is not really possible at the moment in a decent way. But people, of course, try now to see whether they can use similar logics, an RNA molecule recognizing another RNA molecule, and then have an enzyme that modifies the target RNA so that you can do RNA editing. Is this so useful? I don't know. Maybe you want to modify the DNA and then the RNA goes along with it, of course. But there are RNA molecules that have other roles than code proteins. So I think RNA editing is something that a lot of people work on now, using synthetic biology and try to modify RNA. New ideas are also, sometimes you read papers, you say, wow, this is a fantastic idea. Why didn't I have it? I don't know, probably didn't even think about it. Didn't even ask the question of what one could do. So it means we know about DNA, CRISPR. RNA is being done at the moment. So what about proteins? Can you edit proteins? Why don't we work with proteins anyhow? I'm not particularly interested in the DNA. I'm interested in the cell. The cell is made up by proteins, and it's the protein's action that does the job. It's not the DNA action. DNA codes for the protein, and the protein does the job. So why not you look at the business end, at the protein end? Why not target and edit proteins? 20 years ago, you couldn't really edit proteins. Now you can use CRISPR, you can make a different amino acid, so you can kind of edit proteins, but it goes first to the DNA, and you have to make RNA, then the protein has to be made, it takes time, then all the cells have it, it's not very elegant. So now we make a 15-minute break, and then we, 12 minutes, if that's okay with you, until 25. Time for a coffee, and then we expand on from DNA to RNA to protein. And this is a lecture, I'm pretty sure you've heard a bit about CRISPR, but you have not heard about the other things. It took 50 years to edit DNA. We'll see how long it will take to edit proteins in the same manner.